Heidi-ho! Okay, Chapter 7, Section 11. Okay, heat shock response. This is a misnomer. They call it the heat shock response, or heat shock proteins are responsible for this. And a lot of times when you say that, students think this only happens, due to cells getting heated up. No, they came up with this name because, well, originally that's how they found out about these sets of proteins and this set of cell responses. They heated up the cells too much real quickly, boom. And then they noticed that, hey, protein profiles changed. You know, some genes were upregulated, other genes were downregulated. Well, they now know that it's not just heat, that this heat shock response, these proteins can be, yes, from heat, solvent levels change, osmotic stress, UV irradiation, all of these can trigger these heat shock proteins. It's just temperature. That stress was the first one we found that could do this, and we just called them that ever since. These heat shock proteins, known as HSP and then a number designation. HSP 100, HSP 90, HSP 70, HSP 60, HSP 10, 100, the 90, the 60, the 10. I can't remember, that's either the molecular weight or when they just numbering them off because, hey, look, we found these proteins that are, things going on, boom, boom, boom. I don't remember. So don't get hung up on the numbers having a meaning, just that the numbers designate different proteins. And like, HSP 100, this is a set of proteases that are known to degrade proteins, or they form the proteins to make aggregates. Sorry, misspoke there. They degrade proteins, individual, or they degrade aggregate proteins. Because remember, as we've talked about previous section, previous chapters, bacteria overproduce some things, they will just clump them together as a means of storing them away. So yeah, HSP breaks down individual proteins, break down aggregates into individual proteins, which will then be denatured, broken down. HSP 70 is DNA-K. All right, this is a protein that goes and can break things down. Also, upregulates, helps in upregulation, deregulation. GROL and GRO-ES. These are two proteins that are also I do believe these are transcription factors. Okay, let's talk one of these example heat shock responses. Instead of an HSP protein, one of the 5 we talked about in the last slide, we're going to be looking at an alternative sigma factor known as RPOH. Okay, remember sigma factors. We talked about this in previous 2 sections. These sigma factors will control the expression of regulons. Okay, so collection of genes, collection of operons. This RPOH is constantly being made. Thing is, though, instead of having an overabundance in the cell, what we find happens is that it starts to get degraded within minutes of being synthesized. So as soon as translation is done, clock is ticking. Within minutes, it's going to be degraded, no longer functions. But when this stressor occurs, the heat shock, the change in thermal temperature occurs, we see that RPOH degradation stops. RPOH levels will actually increase, and then they start acting as transcription regulators, binding to operons and promoting the upregulation, the increased transcription of certain genes. RPOH is degraded by DNA-K. Okay, DNA-K binds, inactivates RPOH by breaking it down. So just denaturing it to the point where it's inactivated, and then it'll just be recycled. We see that this is one level of control of our RPOH activity in the cell. We see another level of control of activity of RPOH, because remember, it's a transcription activator of operons. Well, it turns out the messenger RNA for RPOH has a tendency to base pair with itself, which interferes with initiation of transcription. So under normal conditions, 32 degrees Celsius for E. coli, RPOH is transcribed. Here's the messenger RNA. Thing is, the messenger RNA starts making secondary two-dimensional and three-dimensional structures, stem loops. These stem loops here, where it binds back on itself, interferes with ribosomes being able to bind and transcribe. The thing is, though, some ribosomes will be able to bind. There will be some translation occurring. Well, as I just said in the last slide, within minutes of RPOH being synthesized by ribosomes, DNA-K binds and starts degrading it. Unless that cell suddenly finds itself in a much warmer environment, 40 degrees Celsius. 40 degrees Celsius is not a whole lot hotter, but, it does make a difference. What happens? Well, quite a few things happen during this shift. As other proteins are being denatured, these native proteins are being denatured because of increased temperature, means loss of tertiary structure down to secondary structure and whatnot. Turns out DNA-K also loses some of its abilities, some of its structure. Means it releases RPOH. So whatever RPOH was there, it's no longer going to be degraded, it's now going to be active. We also look at the messenger RNA that was, bound on itself. Well, because of this increase in temperature, this increase in thermal energy, will cause all of these self-complementary binding to relax. And now ribosomes can bind to it. We then get also an increase in RPOH. Any DNA-K that's present will be bound up by these denatured proteins. RPOH then goes and binds promoters. We now get upregulation of certain genes and certain operons, all in response to increase in the environmental temperature.